So at the end of the post-lecture that I put on CoLab, we finished up some of the regulation and the ways in which you can go back and forth between glucose at the very top of glycolysis or at the end of gluconeogenesis and move all the way through the different steps that we talked about last time, arriving at pyruvate. Today, we're going to move from that point into the next step in metabolism, and accompanying that change to the next step of metabolism, we also have a change in localization. I didn't say this last time, but all of glycolysis occurs in the cytoplasm or in cytoplasmic subcellular assemblies that we haven't talked about. And so there's some organization of glycolysis in the cytoplasm, but for here we'd say all in there. The next step in metabolism downstream of pyruvate moves into this organelle called the mitochondria, which we've talked about in very general terms, but we are going to discuss in more cell biological and mechanistic setting today. I had this slide up to begin because there's a couple key suborganellar structures of the mitochondria that are critically important for metabolism. Mitochondria are double membrane structures. They have an outer membrane and an inner membrane. And by virtue of that outer membrane and the inner membrane, that creates two more compartments within the mitochondria. The innermost region of the mitochondria is called the matrix, mitochondrial matrix. That's where the next step in metabolism that we'll talk about today occurs. In between the two membranes, there's what's called the intermembrane space. You can see that here. And this relates to the second part of aerobic respiration that we're going to get through also today, we hope. And so keep the organization of the mitochondrial matrix and the intermembrane space in mind. And I'm also going to try to note, because regrettably some of the images and figures that I show may have one on the top and one on the bottom, or vice versa. You want to keep track of where ions are flowing or where metabolic steps are occurring. And we're also, hopefully time permitting, talk about other forms of metabolism, notably amino acids and fats that feed into the mitochondria. So it's a real nexus of metabolic activity for the cell. Aerobic respiration. I already said it occurs in the mitochondria, and I already alluded to these two steps that occur in different suborganellar regions of the mitochondria. Now let's give them some names. The first step is, in fact, not a linear pathway like we sketched out for glycolysis. Instead, it's a circle or a cycle called the tricarboxylic acid cycle, or TCA cycle. It has a couple of other synonyms. It can be called the Krebs cycle, named after Sir Edwin Krebs, who discovered it, or the citric acid cycle. And the tricarboxylic and the citric acid will become obvious once we start talking about what's happening at the molecular level in this cycle. The TCA cycle has a couple of roles in metabolism. First is to generate more reducing equivalents, including NADH and another coenzyme that I haven't introduced yet, but we'll talk about today. So more reducing equivalents, which up to this point are mysterious to you, not yet understand where these are useful. Those will come into the second step of aerobic respiration. The other thing, though, I want to say about the TCA cycle is that this is also where amino acids would get metabolized, both catabolized and anabolized in cells. And it's also just downstream of where fats come into play. The second part of aerobic respiration is called oxidative phosphorylation. Oxidative phosphorylation also has a synonym, the electron transport chain, for reasons that also will become obvious. And it's during oxidative phosphorylation, or OXPHOS, that we cash in all of those reducing equivalents. Here we take the electrons back from NADH and this other coenzyme that I'll speak about in a few moments, and then use those electrons to drive protons up an electrochemical gradient between the mitochondrial matrix and that intermembrane space. So there's going to be a relocalization of protons into this intermembrane space. Now you have a proton gradient across that mitochondrial intermembrane. And then by allowing protons to flow down that electrochemical gradient, That's used by a motor protein to synthesize ATP from ADP. That's the grand arc of what we're going to cover today. We're going to begin where we left off. This is a slightly different cartoon of some of the later steps in that transition from glycolysis and into aerobic respiration. All of this occurring in the mitochondria. Whoops. Didn't rehearse all of my timings today. The other coenzyme that I'll just mention by name but show the molecular changes in just a moment that comes into play for the TCA cycle is called FADH2, which goes back and forth between redox states that I'll elaborate on in a moment. But other inputs and outputs into this cycle that are important to keep in mind are water as well as carbon dioxide. Okay, so these are the carbons that get evolved as a result of metabolism. And places where carbon dioxides are released, we already talked about one, this first dehydrogenation reaction of pyruvate to acetyl-CoA. This step releases a carbon dioxide, and then these other two steps release carbon dioxides as well. Those steps, by virtue of releasing the carbon dioxide, are irreversible. We're talking about irreversible steps in glycolysis. Those three, you're not going to take carbon dioxide and put it back into those metabolites. As you might imagine, then, these enzymes are controlled allosterically by different inputs into the TCA cycle. The abundance of ATP versus the abundance of ADP, the availability of NADH, reducing equivalencies of things will impinge on these enzymes. Last time I mentioned just in passing this coenzyme called the Coenzyme A. I want to speak a little bit more about that because this is going to now hand off two of those carbons from pyruvate. Recall one is leaving as carbon dioxide and move it into this other coenzyme that feeds into the TCA cycle. Coenzyme A will come up a couple times today, and the way to think of it is as a means to move two carbon units, all right, so acetyl groups, back and forth onto metabolic substrates or metabolic byproducts, things that you want to break down or things that the cell wants to synthesize. I already said that it gets fed in at the beginning of aerobic respiration. You'll see it again later for fat breakdown and also fat synthesis. There are some things that should look familiar. Another nitrogenous base, ribose sugar. You can see some phosphates in a couple of different positions. Pantothenic acid, you just raise these if you wonder where vitamins and things come from. These will be in multivitamins. They'll be in cell culture media. You need pantothenic acid to be able to synthesize coenzyme A. But the business end of coenzyme A is up here at the top here. It's this sulfohydryl group that is able to pick up two carbon acetyl functionalities reversibly and in an enzyme catalyzed manner. For example, if we have pyruvate dehydrogenase here performing that oxidation reaction that I showed in the preceding slide, what it has is an intermediate where that two carbon unit is loaded onto a thiol of the enzyme itself. That gets then transferred onto the sulfohydryl of coenzyme A, and now you have acetyl-CoA, which is the acetylated form of coenzyme A. That's this step here. All right, so pyruvate dehydrogenase, taking free coenzyme A to make acetyl-CoA, and as a result of this, generating that additional reducing equivalent along the way. All right, so that's an oxidation reaction, and now the electrons are going to be transferred over to NADH and releasing that carbon dioxide, like I said before. What are the fate of those two carbons now? These get fed into the TCA cycle, and the reason why it's called the tricarboxylic acid cycle is that the first recipient of in the TCA cycle is a 3-carboxylic, or the product, after adding in that acetyl group is a tricarboxylic acid. 1 carboxylic acid, 2 carboxylic acids, 3 carboxylic acids, and that product is called citrate. And so before having the addition of that acetyl-CoA, the precursor is another intermediate, which I alluded to in the post-lecture, called oxaloacetate. Oxaloacetate is used in those early steps of gluconeogenesis. It's also the precursor, if you will, to the acetyl-CoA going in and starting the TCA cycle. The projector is not great, but what I want to highlight here, if you can see, there are two CO2s here, and then by addition of this other acetyl-CoA, there's a new two-carbon acetyl group that's been added into the citrate. I just point that out because these pairs of tricarboxylic acids are going to move through the cycle, and some are going to be released as CO2, and others are going to make another cycle around. And somewhat counterintuitive, the CO2s that are released in the TCA cycle, there are two, aren't the two carbons that came in via acetyl-CoA. There are ones that have made it around the loop one or two times. I'll also add that this entry point here, going from oxaloacetate to citrate, is the other irreversible step of the TCA cycle, and also has its own allosteric regulators, NADH, ATP, et cetera. And the reason is once you're in the cycle, you're going all the way. You're going all the way around eventually. All right, so this step here is a water-dependent process. It's going to release free coenzyme A that can be recycled and used for other reactions. We'll keep going around the loop. Akin to glycolysis, I'm not going to emphasize every metabolic intermediate of the TCA cycle. I'm just going to give the highlights and the key points there. This is a good place to refer to what are the blue terms, because these will be the intermediates that I would like you to be familiar with. And the reason is they come up either critically as part of the cycle or they come up in other circumstances. And so it's dual use vocabulary. Around this bend of the TCA cycle, this is the CO2 release and NADH generation loop of the TCA cycle. So we have different intermediates here, enzyme catalyzed. You can see reducing equivalent generated, and here CO2s are removed. The two CO2s that are removed were the two blue carbon dioxides, so the ones that were the old ones in the TCA cycle that get released. The two new ones will eventually become blue as we circle around through there. They eventually get released, but not after they've gone through the loop at least once. So two reducing equivalents, and then there is one generation of a nucleoside triphosphate, but it's not ATP directly. It's actually GTP. But when we do the bookkeeping on the energetic output of the TCA cycle, we're going to count GTP as an ATP. And the reason is that in the cells there are these enzymes called nucleoside triphosphate kinases that can readily take a GTP with an ADP and move the phosphate from the GTP to an ATP. So that exchange can happen very rapidly in cells to meet the metabolic needs. That's the reason why we're counting it as an ATP. On the last turn of the TCA cycle, this is where we generate a new coenzyme that we haven't spoken about before called FADH2. That's the reduced form. And the oxidized form is called FAD. Another electron acceptor, there's going to be a chemical change associated with the gain in those electrons, feeds into oxidative phosphorylation and the electron transport chain, but differently from the way that NADH does. Yeah, so next slide will show this, and then at the final turn here, around when all of the carbon positions have been reset, so to speak, we generate one more NADH, and now we're back to where we were with oxaloacetate in the beginning. And so just thinking about the mass balance here, you had one pair of one acetyl group going in, so two carbons, two CO2s released, so the mass balance is all the same, but along the way generating these reducing equivalents and one ATP through GTP. FAD, let me see what it stands, Flavin, adenine dinucleotide. It's another adenine dinucleotide like NADH. So all this stuff is the same here. What differs is the top part, which is yet another vitamin, riboflavin, I think you've heard about. it can undergo reversible oxidation reduction reactions, chemical changes that occur with this functional group up here, and this is where the electrons are stored and then will be donated to the electron transport chain in the second step I'll talk about. So in its totality, we have a TCA cycle with the following reaction. And we are emphasizing this bookkeeping to figure out where energetic metabolites are being generated along the way, and then how those yield ATP at the final step. So the TCA cycle is coming in with an acetyl-CoA. A lot of these are precursors that at the end will release the 2 carbon dioxides, I said before. We have 3 NADHs, 2 on the first turn and 1 on the back circuit. 1 FADH2, kind of on the middle there, and then the ATP indirectly, or directly, if you will, through GTP. And we have a reminder here that all of this is involved with half of that original glucose molecule, because we were just dealing with one of these pyruvates on the way in. And so all of the bookkeeping on this is ×2 if you want to take the energetic output of a glucose if it's fully metabolized by aerobic conditions. Therefore, where we are up to this point are 4 ATPs net, because we had 2 generated by glucose and then 2 generated indirectly through the GTP to ATP reaction that I described for the TCA cycle. NADHs, we had 2 in glycolysis, 2 in the transition from glycolysis, the entry into the TCA cycle. That's 4, and then 6, 3 times 2 going around through the cycle, and then similar calculations for FADH2. 1 times 2 pyruvates gets you 2. For oxidative phosphorylation is where we convert all of those NADHs and FADH2s to ATP, and you'll see why aerobic respiration is much more efficient at generating ATP than any of the steps that we've talked about up to this point. But before that, I should pause to see do people have questions about TCA cycle. If not, we're going to move on to oxidative phosphorylation, and as I said, it is also known as the electron transport chain. The reason why it's called the electron transport chain is that those electrons that were gained by reducing equivalents like NADH, FADH2, are going to move off of those. They're going to get oxidized, and those electrons are going to move into a series of complexes in that mitochondrial inner membrane. And as a result of gaining and losing those electrons, they get propagated through a number of complexes in that mitochondrial inner membrane. They're going to undergo conformational changes that allow protons to be moved concurrent with those conformational changes that occur in a redox-dependent manner. We have complex 1, 2, which is not shown here, 3, and 4. And then at the end, we're going to talk about the pump that will leverage the proton gradient that I introduced a couple of slides ago to synthesize ATP. But first, we need to talk about how those protons get pumped in the beginning. Oh, and then here's my reminder. TCA cycle happening on this side. Matrix on the bottom, all right, intermembrane space on the top. The first thing we will do is zoom in on what's called complex 1. And the boundary that I drew around there is a more accurate description of what fully complex 1 was. And the reason why we know this now is because the crystal structure of complex 1 was in fact determined only about 5 years ago. So I have a better understanding of how some of these other steps are involved. This is a massive complex, okay? This is a crystallographic nightmare. I don't know how they did it. But it's really empowering because now we can understand from a structural perspective how redox changes in this collection of enzymes translates to the ability to pump protons from one side of the membrane to another. There's a whole lot of details in this cartoon. I just want to give you the gestalt of what's happening. First, I want to orient you. Everything's flipped now. Matrix is on the top. Intermembrane space is on the bottom. So we have the matrix up here. The first step that occurs, this is the transfer of electrons from NADH only, onto another coenzyme that's bound to complex 1 called Flavin mononucleotide. So Flavin, if the electrons leave NADH, it converts back to NAD. Those electrons go on to the Flavin mononucleotide FMNH2. Now what occurs is that Flavin mononucleotide, through different intramolecular electron transfers, will propagate those electrons through a number of there's different iron sulfur clusters and things that can undergo reduction oxidation states very easily. And as it moves its way down through complex 1, it will get into this center portion of complex 1 that's going to give rise to a coordinated change of the entire complex, depending upon where those electrons are in the chain. So what occurs is that once you get down to here, and the other thing I should say is that this is where coenzyme Q is another redox-dependent electron state changing coenzyme that's embedded into complex 1. Once it gets down into this part of complex 1, there are 4 cavities here that open either on the intermembrane space side or on the matrix side that can bind or release protons depending upon their redox state. And so what occurs is that when the complex 1 is in this state here, it's able to release protons that it has previously bound into the intermembrane space. And conversely, when it's in this alternative state, it's binding protons from the mitochondrial matrix. So what you have is a flipping back and forth of binding protons on one side in one conformation, depending on its redox state, and then flipping over to the other side to release them into the intermembrane space. The thinking is that the same type of conformational and redox-dependent changes occur with all of the other complexes, but at my last look, they haven't been determined in as great structural detail. As those electrons move back and forth in this complex, they get released from complex 1 and continue to percolate through these additional complexes. There are different cytochromes involved. You don't have to worry about the details of these, but the idea of conformational changes and protons being transferred from the matrix into the intermembrane space are the same, but the numbers on them vary. Complex 1 will move 4 protons, as you saw by its cavity. The next one is just complex 3, skipping over number 2. 2 comes in another place. 3 will also move 4, and then complex 4 moves 2. So 4 plus 4 plus 2 is 10. At the very end, and this is a complex 4-dependent process, if you'll recall back in the beginning when I talked about aerobic respiration, what is the terminal electron acceptor? That's where the oxygen comes into play. So oxygen receiving those electrons from complex 4, okay, and getting reduced to water, that's the end of the electron transport chain. The net result is for 1 NADH, we've now moved 10 protons from the mitochondrial matrix to the intermembrane space. What happens to FADH2? This is where complex 2 comes into play. Complex 2 is another electron acceptor specific to FADH2, but in contrast to complex 1, which moves 4 protons, complex 2 moves 0. So it can accept the electrons, but it won't be able to pump any protons as a result of conformational change. The net result then of an FADH2 is rather than generating a proton gradient of 10 protons across the membrane, it only generates a gradient of 6. And now why the heck do we need all these protons? will do all the accounting on how many protons and things get moved. But as you have these protons and as you have this electrochemical gradient between the mitochondrial matrix and the intermembrane space, there is a particular pump that is remarkable, and that is really a pump run in reverse. You guys covered pumps with Professor Barker, right? The way that they ordinarily work is that they'll hydrolyze ATP to drive solutes up a concentration gradient, right? This pump, called the F1, F0 ATP synthase, or ATP ACE, is a pump running in reverse. You guys aren't homeowners yet, but if you have like a heat pump, you can run it as a heater, or you run it in reverse and it becomes an air conditioner. Okay, this is the same type of thing. So rather than having a pump that hydrolyzes ATP to move up a concentration gradient, it allows the protons to flow down their concentration gradient and then synthesizes ATP in the process. And it really looks like a generator. It has a stalk, a series of subunits here, and as the protons flow down through that electrochemical gradient, there are 120-degree shifts in that almost like turbine as it goes around, and each time it moves that 120-degree shift, one ADP is converted into an ATP with an inorganic phosphate. And so 9 protons going through. Oh, wait, I had 2 questions. I probably missed it since it spoke. Oh, I see. Thank you. What side is which side going on here? So here, intermembrane space on the bottom now, and then matrix on the top. Thank you for asking that. Yeah, so what's happening is that you've already pumped, it's already going up, and then now this pump is going to be open, the gradient is high enough, and this is going to be spinning like a wheel as protons are going through from the bottom up to the top. ATP synthesis happening in the matrix. There was another question in the back. Oh, same one, thank you. This is the one slide I didn't have the note to remind on these things, so I'm happy that you asked. Therefore, if you do a 360-degree rotation, that is 10 protons down the gradient and 3 ATP molecules synthesized. So let's do a couple numbers here, just to review what I already told you. 10 protons per NADH, 6 protons per FADH2, if they're used to their full capacity, and 3 of them get used up, if you will, going back down the gradient that was used by bringing them into the electron transport chain to synthesize an ATP. Therefore, let's go back to glucose. Remember, we had 10 NADHs if you fully metabolize glucose all the way through aerobic respiration. 10 NADHs fully through the electron transport chain, that should be 100 protons pumped. So if we take 100 protons pumped and we divide it by 3, that's going to give us, well, it's going to give us officially 33.333 ATPs. You need a finite number of molecules. But the reason why we have here approximately 30 ATPs is that the conversion of those protons into ATP synthesis from the reducing equivalent of NADH is not perfect. There are leaks along the way. We're going to talk about the leaks along the way next class. But this is why we assume maybe a 10% loss of chemical energy along that electron transport chain, which is why we get the number of 30. FADH2, we can do the same thing. So that, recall you have 6 times 2 is 12, and then just for ease, we're going to divide that 1 by 3 and give 4. Thus, at the end of it all, if we have fully converted all of the NADHs, FADH2s with those approximations like I had said before, combining with the 2 ATPs that are generated via GTP and the TCA cycle, combined with the 2 ATPs that were generated by substrate level phosphorylation during glycolysis, we get at the number that metabolism aficionados know to heart, which is that 38 ATP molecules per molecule of glucose can be generated under aerobic conditions. I'd like to emphasize that 34 of them are via the electron transport chain and oxidative phosphorylation. That's why aerobic respiration is preferred over anaerobic fermentation. And that's how we arrive at the complete reaction, which is taking 1 glucose molecule, evolving 6 carbon dioxides and generating 38 ATPs with water and organic phosphates on the balance. How's everybody doing so far? I'd like to say that that's it with metabolism. There are a couple of other things that I'd like to touch on related to organelle transport, amino acids, as well as fats, because then what we're going to use those pieces of information for is thread them together back into diets and different relationships between these different metabolic pathways that I touched on before. So to begin, transport. In the very first cartoon slide that I began today's lecture with, we showed how pyruvate needs to get from the cytoplasm into the mitochondrial matrix to be able to be oxidized and go into the TCA cycle. How does it get through there? It is a I mean, there are carbons in there, but it also has polar constituents, and it has to go through 2 membranes to be able to get into the mitochondrial matrix. Those other metabolic intermediates of the TCA cycle, I didn't emphasize them, but we talked about citrate, oxaloacetate, malate, et cetera. These all need dedicated transporters to move them. And so this is what there are. They're either symporters, antiporters, different channels that will now allow these metabolites to move in and out of the matrix. And this is important because they are charged species that won't just passively diffuse through. Yes? Is the same setup true for the outer mitochondria? Yeah, so it's shown in just cartoon form here for one of the membranes, but the same thermodynamic argument holds for the outer membrane as well. And as far as I know, like the actual dicarboxylic acid transporters or tricarboxylic acid transporters are the same for both of them. Some of them are synthesized by mitochondrial DNA, but others are localized. This is where mitochondrial targeting sequences and things like that come into play. All right, so let's move beyond the metabolism of glucose and talk about other biomolecules. What happens with amino acids? This is an interesting facet of the TCA cycle, in that many of those metabolic intermediates that I de-emphasized in the cycle are very close to a number of different amino acids. As an example, we can take alanine as just a regular amino acid and through a transamination reaction, convert this to a carboxylic acid, and now we have pyruvate. We can take aspartate here and do the same trick, and then now all of a sudden we have oxaloacetate. And if you wonder, well, where are those amines and where are those carboxylic acids going and coming from? They're usually coming from this TCA intermediate alpha-ketoglutarate turning into glutamate. And so there are these enzyme catalyzed reactions that will swap an alanine to a pyruvate and then a ketoglutarate to a glutamate. And these are means by which the cell can generate what are called non-essential amino acids. We don't need to consume those in our diet because very quickly by these different reactions related to the TCA cycle, we can synthesize de novo these amino acids. This is not true for all amino acids. Essential amino acids, ones we need to take via our diet because we can't synthesize them. Examples are like histidine, phenylalanine, tryptophan, et cetera. And so very close relationship between amino acid synthesis and the TCA cycle. Fats are also closely related to TCA cycle, but much closer to the entry point, acetyl-CoA. Fats go through, I provide the complete chemical reaction here for your information. Talk about a couple of the steps, but I'm going to work through it at a high level. We're going to start with fat catabolism, so the breakdown of a fatty acid. All right, so the way that this works, we're going to start this, actually the first step is hooking it up with a CoA molecule. All right, so rather than an acetyl-CoA, it's a CoA tied to the big fatty acid chain. There's a dehydrogenation reaction. You're familiar with this coenzyme, the reducing equivalent that's prepared. We have a hydrolysis event, and then another oxidation event to create this double bonded carbon here at the beta position of the fatty acid. So this was the CoA molecule, carboxylic acid, alpha and beta. What occurs at the final step here is, this is where the name of beta oxidation comes in, is there is this enzyme catalyzed reaction at this beta position that cuts off two of the carbon molecules, generating an acetyl-CoA. And so through redox-dependent processes, fatty acids get broken down 2 carbons at a time from the carboxylic acid position, moving through the aliphatic, generating acetyl-CoA molecules along the way. And so that's the essence of beta oxidation. Therefore, we can now start putting together fat metabolism, glucose metabolism, TCA cycle together based on inputs and outputs, the diets and the things that we eat, and also the way that our body handles fats in our bloodstream. I mentioned this at the beginning when we were talking about obesity and high levels of triglycerides circulating. What are triglycerides? Chemically, they're not all that different from phospholipids. You remember cell membranes, 2 fatty acid acyl chains, and then there'd ordinarily be a polar head group here to make a phospholipid that would go into a membrane. Triglycerides, what they basically have done is substituted that polar head group with another fatty acyl chain. 3 fatty acyl chains and then a glycerol backbone holding them all together. These are the biomolecules that will get stored in our adipose tissue and fat depots, et cetera. These are also the things that circulate in the blood, transported by things like lipoproteins to be able to deliver energy to target tissues. When triglycerides are broken down, are catabolized, it occurs here by these ester bonds here to generate free fatty acids. The free fatty acids get metabolized by beta oxidation, generating acetyl-CoA molecules. Those acetyl-CoA molecules can go one of 2 paths. I'm going to only talk about one. Well, I already did talk about one today, which is the acetyl-CoA going into the TCA cycle to generate reducing equivalents, et cetera, from there. So rather than coming in off of the oxidation of a pyruvate molecule, you can have acetyl-CoA coming in from the beta oxidation of fats. That's where we have this arrow drawn here. After the free fatty acids have been cleaved off, what happens to the glycerol backbone? There's a phosphorylation of glycerol that occurs, followed by another oxidation event. And now you're at glyceraldehyde 3-phosphate. Recall that glyceraldehyde 3-phosphate is one of those intermediates. Somewhere in the middle, this is probably not the best drawing, of glycolysis. Right before that oxidation event, the dehydrogenation event, that gives you the reducing equivalents during glycolysis. So when you have fats going in here, they feed metabolites into the middle of glycolysis and then at the entry point of the TCA cycle. And as I had sketched out before, amino acids through those transamination reactions and so forth go in via oxaloacetate or other derivatives here into the TCA cycle. This alternative path here of excess acetyl-CoA we'll talk about at the next time, which occurs when you have deficiency of some of the intermediates in the TCA cycle, and this is where we'll relate to different diet fads and things along those lines. Everybody's very quiet or tired today. I finished well ahead of time. Okay, so your question is the thing I just alluded to at the very end. I said, well, what do you mean by a deficiency? Yeah, and so is that correct, what your question is? Yeah, so recall that every single one of those acetyl-CoAs that goes in needs to have an oxaloacetate to accept it to become a citrate molecule. And therefore, you're right, it boils down to the relative stoichiometries of how many oxaloacetates do you have relative to the acetyl-CoAs that are being produced to determine the sufficiency of it. And that's what the theme is going to be of the next lecture, about how interconnected each one of these metabolites are. You take away from one place, and then things back up somewhere else or get deficient elsewhere, and they lead to these compensatory changes that end up not always generating the effect that's desired by dietary intervention. Mm-hmm. Oh, I see. Your question, you're asking about like the redox environment of it? You can make inference about the pH, right, of some of them. I'm thinking about this on the fly. No one's asked this question before. So so this is like a quiz question for me. I don't know the answer. I'm going to figure it out right now. In the matrix, recall that you're having a lot of electrons being donated from NADH into those complexes, right? And so I would take that as an oxidizing environment, because all of those reducing equivalents are transferring electrons into the matrix. For the intermembrane space, I can't say, because really what you typically associate that is with that concentration of protons, and there isn't really any redox reactions that I'm aware that are at least critically involved in the extent that I know metabolism. And so that one is an unknown quantity. I'm sure it is known, but I don't know it. Okay, I guess I'll see you. No, you keep going. That's fine. I will call it. I'm going to stand here and stare at one another for 15 minutes.
That breakdown of fats, that pathway, it still starts at glucose on the
So your question is the starting, how do you interpret this diagram? I haven't fully explained it. The bidirectional arrows here emphasize none of these steps are irreversible. So depending upon the concentration of products and reactants, you can run these in either direction. What I introduced on this slide is if you're overloaded with triglycerides, if you have a lot here, how will that flow into the other pathways? But next time when we chat, we're going to talk about ways in which if you eat a lot of this, but not this, you take this away, you add more, how do those then put pressure points? Because none of those steps, they can go in both directions depending upon the availability of substrates. Okay, I'll see you on Saturday.